Hello everyone and welcome. I'm Carla Redlinghuis from the Department of Genetics here at Stellenbosch University within the Faculty of AgriSciences.
Hi, and I'm Nathan Lacordia, also from the Department of Genetics.
In this short video, we'll introduce you to what genetics is, the opportunities our department has to offer you, and what a typical day looks like for a postgraduate student within the department. Before we talk about what genetics is today, it's worth briefly looking at where our department comes from. The Department of Genetics at Stellenbosch University has a history that dates back to 1898, making it one of the long-standing contributors to scientific teaching and research at the university. The department formally emerged from the Department of Agricultural Botany in 1925 and has since expanded beyond its early agricultural roots, continuously adapting to new discoveries and technologies.
That legacy continues today. Located in the J.C. Smuts building, the Genetics Department, together with its sister facility, the Institute of Plant Biotechnology and Breeding, the IPB, in the Natural Science Building, works across multiple fields such as systems genetics, genetic modification, population genetics, biotechnology, bioinformatics, and advanced plant breeding. So, let's start with the obvious question. What is genetics? Genetics is the study of genes, hereditary, and genetic variation in living organisms. It helps us understand how traits are passed from one generation to the next, and how DNA shapes life itself.
To study genetics effectively, we use a combination of molecular and biotechnological techniques, together with computational tools such as bioinformatics and machine learning. These approaches allow us to identify genes, understand how they function, and explore how they influence traits in plants, animals, and humans.
Genetics is also a rapidly evolving field, staying current with new research throughout scientific literature, conferences, and professional networks is an essential part of being a geneticist.
So why is genetics important? In our department, this comes down to 4 key areas.
First, genetics plays a crucial role in understanding diseases and health, from viral infections to vaccine development and human health more broadly. While we don't focus exclusively on human health, topics such as viral genetics and emerging diseases are integrated into our academic framework.
Second, genetics is central to pharmaceutical science and personalized medicine. Our human genetics laboratory, for example, focuses on mental health disorders such as schizophrenia and OCD, as well as pharmacogenetics, helping us understand why individuals respond differently to medications.
Third, genetics is essential for agriculture and food safety. In South Africa, climate stress and heat waves pose real challenges for farmers. We have several labs in the genetics department that address these challenges by working on crop improvement, like the Cereal Genomics Lab, Citrus Biotechnology, and the Fruit Functional Genomics Lab. But we also have a larger research environment known as the IPB. These labs are working on developing plants that are more resilient to harsh environmental conditions, but also improve nutritional quality of different crops as we achieve this by advanced plant breeding, but also use molecular tools like CRISPR.
Finally, genetics plays a major role in conservation and environmental sustainability. One of our research groups focuses on population genetics in endangered and marine species, including shark and abalone populations. This research informs conservation planning, species management, and long-term biodiversity protection.
We also have the Forestry Genetics Laboratory, which focuses on sustainable forestry and ecosystem management. By applying genetic and statistical tools to forestry systems, this research supports natural resource conservation, while addressing challenges linked to climate change, land use, and long-term ecosystem resilience.
As mentioned earlier, to study genetics effectively, we use a combination of molecular and biotechnological techniques together with computational tools. These techniques overlap in almost every postgraduate project, whether at honors, master's, or doctorate level.
Hands-on lab work, or what we call wet lab work, refers to the experiments performed on the lab bench. This includes techniques such as DNA and RNA extraction, as well as manipulating genetic material using molecular tools. A well-known example is the production of insulin. Researchers synthesized the DNA sequence of the insulin A and B chains and inserted them into bacteria. These bacteria were given the instructions to produce insulin.
Another example of genome editing is the use of CRISPR. In the viticulture lab, this technique is often used to silence specific genes by creating targeted cuts in DNA and then evaluating the effects in tissue culture or in the field.
In contrast, dry lab takes place away from the lab bench. This includes writing up experiments and findings, as well as using computational tools to analyze data that was generated through the wet lab work. One prominent example I can make is the use of a genome assembly process, which is essentially like building and solving a giant puzzle. And then by analyzing this puzzle, so to speak, we start identifying genes linked to genes of interest.
At this point, you might be wondering whether genetics is the right field for you. We usually encourage students to consider a few questions.
Do you enjoy biology and problem solving? Are you curious and interested in discovery? Are you comfortable with working with large data sets and numbers? Can you think critically and creatively? Are you prepared to work hard? If you have answered yes to any of these questions, genetics could be a good fit for you.
Genetics is a major teaching and research discipline at Stellenbosch University. As a result, the department has access to a wide range of modern equipment and technologies. For example, once we isolate a gene in the lab, we often confirm and analyze it using specialized software linked to high-end molecular equipment.
Research in the department spans plants, animals, and human genetics. This includes drone technology for precision plant breeding, advanced analytical tools in human genetics for personalized medicine, and population genetics research in animals with applications in conservation and forensics.
Here is a video of an average day of a postgraduate student in the shark and ray lab. Most of our time is spent in the laboratory, and our project focuses on species distribution and genetic differences between populations. My day-to-day involves planning experiments, extracting DNA from samples, running molecular assays like PCRs, and analyzing the results. Lab work can be time-consuming and requires patience. Some experiments take days or even weeks to complete. But it's incredibly rewarding when everything comes together and you see how your data contributes to answering bigger research questions about marine biodiversity and conservation. We also make sure to maintain a healthy work-life balance with time for activities outside the lab.
For my PhD, I work mostly in the bioinformatics office in the genetics department. My project involves the sequencing of the genome of a grapevine to identify genes associated with disease resistance. This requires a lot of computational work and statistical analyses, but it can be very time-consuming. However, one of the major advantages of my project is that it allows me a lot of flexible free time. So I can do what I do from the office, from the gym, from home. So at the end of my PhD, I'm hoping that my research as a whole would contribute to the establishment of virus-resistant grapevines so that the production wouldn't suffer so much from the losses that occur because of these diseases.
There are several pathways into genetics depending on your interests. If you're interested in human genetics, you would typically begin with a BSc in Human Life Sciences or Molecular Biology and Biotechnology within the Faculty of Natural Sciences.
For animal genetics, students usually enter through programs such as Animal Production Systems in the Faculty of AgriScience. For plant genetics, students usually study plants and soil science, also within the Faculty of AgriScience. All of these routes can lead to postgraduate specialization within genetics department by selecting genetics as a major in the second and final years of study.
Because the Genetics Department and IPBB are teaching departments, our work combines teaching, research, and student supervision. Students encounter genetics teaching from first year through to advanced undergraduate modules and postgraduate research. Because teaching and research are closely linked, students are exposed to current scientific questions and real research environments throughout their studies. To conclude, genetics is a highly integrated and collaborative field. We work with engineers, clinicians, physiologists, ecologists, and many other disciplines to address complex biological problems.
So thank you for taking the time to learn more about the genetics department. We wish you every success in your studies and hope to see you soon, hopefully in the first year class.